Hello everyone, how are you? My name is Ujwala and welcome to our channel Science. Today, let us learn about the basics of PCR, that is polymerase chain reaction. Here the PCR is specially working to amplify the DNA molecule. If you are new in our channel, please subscribe our channel and don't forget to like. Let us learn first what is PCR. The full form of PCR is polymerase chain reaction. PCR is a technique used in the lab to make millions of copies of a particular section of DNA. So actually, PCR used to amplify and multiply the DNA molecule. Whenever we have a small amount of DNA and we need to be more amount of DNA for some examination or identification purpose, we have to multiply the DNA. So here we can use PCR. It was first developed in 1980s. The polymerase chain reaction, that is our PCR, was originally developed in 1983 by the American biochemist named Kary Mullis. Here, he was also awarded the Nobel Prize in Chemistry in 1993 for his pioneering work of the PCR, which helped the world of DNA in a betterment. Now, the PCR is used in molecular biology to make many copies of amplifying DNA, where the small section of DNA or a gene can be used. Here, majorly the 5 core ingredients are required to set up a PCR. So let us see first which are these 5 main ingredients. The first thing, that is the DNA itself. It is called a template to be copied, because first we have to have some DNA molecule or a strand of small molecule of DNA, with the help of which we can multiply and duplicate multiple copies. So DNA is the primary requirement, and here it is called a template. Secondly, the primers. Here the primer is a short stretches of DNA that initiate the PCR reaction. As you can see, they can bind with the DNA at some complementary region, and from where the DNA amplification can be started. The design to bind to either side of the section of DNA you want to copy. So both the strand of DNA can be take up the primer in them, and then after the multiplication process will be started. The third required element is also known as DNTPs, that is nitrophosphates. Here the DNA bases, that is as you can see, A, C, G and T, that is thymine, cytosine, adenine and guanine. These are the building blocks of DNA and are needed to construct the new strand of DNA. As we know, we need to construct the whole new molecule of DNA, where we need some raw material. So here these all building blocks are arranged in such a manner on the DNA, and then after the new DNA molecule will be formed. The fourth required element is a tag polymerase enzyme, where to add in a new DNA basis. This tag polymerase enzyme is very much necessary, because with the help of this enzyme only the DNTPs, what we just have discussed, can be bind with the DNA with a proper functioning. And the fifth one is a buffer to ensure the right conditions for the reaction, because we have to maintain the right condition, the temperature and the pH, then only the DNA can be multiplied. Here all the 5 elements will be come together, and here the PCR involve a process of heating and cooling called thermal cycling, which is carried out by the machine, and the machine name is thermocycler, which generally maintain the temperature required in the various stages of the PCR. Now there are main 3 stages in the PCR process. Let us learn each one in detail. First, we'll see the overview. In the first stage, the copy of DNA will be denatured. With the help of high heat, it will be aparted into two different strands. Then after, in the second stage, that is annealing, the primer will be attached, and in the third stage, the multiplication process will be moved forward. So let us learn each sample and each stage in a detail. The first stage, named denaturing. As per its name, it denatures the DNA. So when the double-stranded template DNA is heated to separate it into two single-stranded. As we can see over here, a complete DNA in a high temperature condition divided and apart in a two separate DNA strands. In the second stage, that is annealing, when the temperature is lower as compared to the denaturing state, it is to enable the DNA primer to attach to a template DNA. So here the primer can be attached at the complementary region. The primer is nothing else than a small molecule of DNA. This will be attached over here, from where it gives the place to initiate the DNA amplification. The third stage is extending or the extension stage. When the temperature is raised and the new strand of DNA is made by the Taq polymerase enzyme. As we have already discussed, the Taq polymerase enzyme is a specialized enzyme to maintain the smooth functioning of the DNA amplification. Here the green colored part is the DNA polymerase. So here one by one the DNA NTPs will come here and bind to the DNA, and this will be supervised and maintained with the help of Taq polymerase enzyme only. So this is how all the three steps can be carried out. So here we just see the overview that the double-stranded DNA first in the primary stage of denaturing allow the temperature from 94 to 95 degrees Celsius, where the DNA will be aparted into two separate strands. Then only the further process can be started. In second stage of annealing, the temperature allowed is 50 to 56 degrees Celsius, where this lower temperature favors the primer binding. And the third stage, that is the extending stage or the extension stage, with a 72 degrees Celsius temperature, allows various dNTP blocks to attach the DNA. So whole DNA will be prepared and be ready over here. So this is how all the steps will be carried forward. So first step is denaturing at 98 degree approximately Celsius, which gives DNA a two strands separate. Then after, in the second step of annealing, 50 to 60 degrees Celsius allows primer to bind easily. The primer are kind of a primary functioning which allow the further binding of the dNTPs. In third step of extension at 72 degrees Celsius, with the help of Taq polymerase enzyme, the further preparation and the binding of other dNTPs will be carried out. And this is how two separate and independent DNA can be created. So this is how from one DNA we now have two DNA. Afterwards, these three stages are repeated 20 to 40 times as per our requirement of the cycles of the DNA number. Here it doubling the number of DNA copies each time, because here in a one cycle we had one DNA, but at the end we got two DNA. If we further repeat the cycle, we will get 4 DNA from the 2 DNA. Then after it will be multiplied into 2, and every time after every cycle we will double the number. So this is how the older DNA will be released and make two strands independently, and from where another blocks will be bind and form a new DNA strand. So from one DNA, that is called parent DNA or the template DNA, will be released into two strands and the new strand will be formed. Here the older template, it is called the template DNA or the parent DNA, and whatever the new strand is created called the daughter DNA. Here, because the daughter DNA is totally dependent on the parent DNA, it is called a daughter one. So this is how after one cycle we will get double number of DNA. So from first cycle we will get 2 from 1, then in second cycle 2 to 3, then after the fourth number, in third cycle 4 to 8 DNA, in fourth cycle 8 to 16, then from 16 to 32, then from 32 to 64. Likewise, it will be increase in number in multiplying of 2. If we require whatever number of DNA copies, that much amount of cycle we have to run over here. Then this is how we will get large number of DNA copies as per our requirement. So this is all about the PCR, how the DNA will be introduced with the help of variety of ingredient, and with the help of all these parts, the 5 different parts what we have discussed, the DNA will be multiplied into double number. Thank you very much. Please subscribe our channel, hit the bell icon, and if you are new in the channel, don't forget to like and subscribe. Thank you.